But to introduce this session one, welcome Oumeya Adjali. Oumeya, okay, you can come, thank you. You're from INSERM, and you're going to present gene therapy using AAV viral vectors from vector engineering to preclinical evaluation. Thank you.
So, good morning, everyone. I'm very delighted to be here. I want to thank the organizers for giving me the opportunity as an INSERM research director, so academia, to talk about gene therapy and AAV vectors and where the field is standing on and what are the challenges we're facing to cure patients. So, okay. So, as you may know, AAV viral vectors are widely used today for gene transfer. They are almost the most used vectors in gene therapy, especially for in vivo gene transfer, where they represent about 85% of the used viral vectors. And this is a study published by Map Design about one year and a half. So what are these AAV vectors? So they are derived from a parvovirus family, and they have a number of advantages to transform them in viral vectors for gene transfer. They are derived from viruses that are non-pathogenic or not associated to pathologies, and they are a dependovirus, which means that they are defective in replication alone and that they need dependovirus. So this is like a safety feature very important for gene therapy. One major advantage is that one unique single in vivo administration can allow very long gene transfer, and we have really years and years of follow-up, even in patients today, without major integration. So they remain mainly in episomal forms, despite the fact that now we know that we have some integrative events also, but no associated to oncogenesis so far. One limit now is that they are very small, these vectors, and the encapsulation size is very limited to only 4.7 kilobases, which means that they are not today, as they are, they cannot be used for large genes. But I will talk on that later on. So what is great with AAV vector is that we have plenty of natural serotypes and hundreds of variants now in the labs and in the industry, which means that we can choose à la carte, let's say, one serotype or one variant, playing also with the mode of administration to target virtually all the organs. So for instance, if you want to target the brain, we can use CNS, intrathecal, intracisternal route administration, and AV10 or AV9. If we want to target the muscle, we can use the intrasystemic route of delivery using AV8 and AV9, which are musculotropic and cardiac tropic. And actually, the AAV vectors are already in the market, as you may know. To date, we have, 7 products have already been approved. One was withdrawn, the Glibera, the first one, approved. So these are the products approved by either the FDA and or the EMA for ocular, muscular, metabolic disease, as well as hemophilia. The 2 last ones approved in Europe are for hemophilia A and hemophilia B. But those vectors work using very high doses. And this is very true for systemic or intravenous administration. So for instance, for those products, the 2 last ones for hemophilia and the approved product for Duchenne myopathy, LVDs, we need between 10 to the 13 to 10 to the 14 viral genome or virus, recombinant virus per kg, per kilo, which is very huge. And that means that there is still a gap between, let's say, preclinical developments, manufacturing, and the use of these vectors in patients, because there is clearly a lack of specificity and efficacy of these vectors, and we need really to work on that to reduce the doses, to reduce the needs of manufacturing and the cost, of course, because the cost is very high, is more than 1 to 3 million per product approved today in the market, to reduce as well the toxicity and immunogenicity issues. And there are two other major limitations. The limit of the encapsulation size, I was talking earlier, which means that we also figure out how we can treat disease where large gene, larger than this size, are affected or are the targets. And the other one is that the animals, the model animals we have used so far, mice, non-human primates, have not always predicted what happened in patients. So we really need to also continue developing new models of disease and new models for vector screening and evaluation. So, yeah, so I think I can skip this one because I have. So in my lab, target for translational research in gene therapy in Nantes, we are working on some of these limitations and developments. I can say that the lab is really in the heart of the objectives of the National Initiative Stratégie d'accélération des biothérapies et bioproduction. The fact is that we're gathering in the same lab, in the same site, scientists from different fields. So we really believe that multidisciplinarity is needed in the field of gene therapy to develop next gene vectors. And so for that, we have not only biologists, but we have also process engineers, bioinformaticians, chemists, biophysicians, immunologists, and this allows really us to cover the whole chain of AAV vector development, from vector design in silico to vector engineering, vector screening and evaluation in preclinical models, process developments to produce those vectors in large scales and move to the patients. And to make the translation possible to patients, we have also been pioneer to certify the whole activities of the lab, including research activities, under the ISO 9001 system to make the data robust for regulatory agencies and pre-INDs and INDs. And so regarding the manufacturing part, we have a facility in the lab called Centre de Production des Vecteurs, which is one among the 8 integrators of the stratégie d'accélération des biothérapies. So we believe that it's very important to, of course, deal with the process itself. So systems of purifications, but we also try to back to really fundamental biology to the cell that is producing AAV vectors, because here again there are some innovations to make on these cells to increase the yield of production and increase the quality of AAV vectors. But now, during the remaining few minutes, I want to talk about the other hurdles, much more vector engineering and preclinical. So for vector engineering, the majority of people have worked, and we are also working in some projects, on molecular vector engineering, which means engineering of the capsid or engineering of the viral genome itself. But one other possibility is to combine different fields, to combine chemistry and biology, and try to generate chemically modified vectors where we use chemistry methods to conjugate ligands, selected, of course, to target a cell or an organ of interest. So we have been working with those methods. We have a chemist, Mathieu Mével, in the lab, working with a chemistry lab in Nantes, academic lab, And we have already provided some proof of concept to better target than native AAV, the ocular, the retina, the liver, the muscle. Now we are working on the joints to treat maybe one day arthritis, and also the CNS, central nervous system. So another thing is that we not only need vector engineering, but we also need methods to characterize those vectors and correlate the efficacy of these new vectors to their safety, the absence of toxicity, but also efficacy. And to do so, we really need to move from the conventional methods, analytical methods used so far, so the qPCR targeting only one very short sequence in the viral genome, to high-throughput sequencing methods that enable a full characterization of the integrity of the viral genome, because this is really also a limit with the manufacturing methods we have today, and also to characterize or look at the DNA contaminants that we will have certainly from the cells that are producing the AAV vectors. So we have been, again, one among the pioneer labs to develop a DNA-seq method to characterize different batches produced from different methods, and we can even compare and have a signature from the different methods and correlate the quality of the batches to safety and efficacy. Now, regarding the preclinical evaluation, there's also still a gap, because the mouse models, well, they are very good to evaluate the efficacy of a vector, but they don't at all predict immunogenicity. This is what we have seen upon translation to large animal models and patients. And as you know, also non-human primates are now very difficult to use and not very ethically possible to use. And so we believe that moving to other animals could be one possibility, one avenue. So, for instance, we have been working in Nantes at the development of transgenic rats model to modelize Duchenne myopathy and also Stargardt disease, which is a macular-ocular genetic disease. And these models, above the mouse model, they have also an advantage is to make possible the evaluation of the surgical methods of route of administration more closely to a large animal model than the mouse models. Another possibility, for instance, is to move from large non-human primate model to the pig and the mini pig. And the mini pig is very promising, looks as very promising for the evaluation of ocular gene therapy products, because it has a retina that is not so far, let's say, from the human retina. The last possibility is to generate humanized mouse models, where we can humanize either one organ or multiple organs. And it's very interesting to evaluate the immunogenicity of AAV vectors upon adoptive transfer of human, for instance, cells with pre-existing immunity against AAV. Now, we know that animal models are not perfect, and they need additional complementary models. And we are working also, and many others, in the field of gene therapy and AAV gene therapy on human organoids using iPS cells from healthy and patients. So these are very interesting because they will have the exactly same genetic background. Maybe not the epigenetic, this is another story, but at least we can have the mutation we want to treat. And of course, those models are not yet fully mature, and there is still a lot of work on this, but they look really promising to screen AAV variants. So last direction is how to overcome this limited capacity of encapsulation. So there are different methods. Some methods or some technology are used dual vectors, which means that we can use two vectors with half and half gene, Or we can also insert some specific sequences and express half-and-half protein and make the recombination possible in vivo. But another possibility is to combine next gene therapy strategies. For instance, an engineered vector with increased efficacy to gene editing CRISPR-Cas9 strategies, and I know that there will be some gene editing also discussions. So, using CRISPR-Cas9 not to correct a point mutation, but using it with the AAV vector to bring and replace a part of the gene where a hotspot of mutation is observed. And we are working within the frame of peer-to-peer funding with great partners on these strategies and with the proof of concept for against Stargardt disease, the macular genetic disease. So to conclude, so AAV vectors are really promising. They are on the market. They are treating dramatically diseases such as SMA, for instance. But there are many, many developments yet to do to improve the efficacy, to improve the safety, to reduce the doses and reduce the manufacturing needs. And of course, we need also in parallel to work on much more relevant models to evaluate and screen the new variants, which means that we must go from bench to bedside and back again every time to develop a new next generation of gene therapy vectors. And it is clear that the partnerships, the partnerships between public and private can only foster and accelerate those developments. So I am looking forward to the discussions I will have during these 2 days. I've just, if I can have 10 seconds, just to, as a, let's say a follow-up of these hybrid days, grid days, I just want to ask you to save the date because as a member of the board of the French Society of Cell and Gene Therapy, I'm the vice president of the society, and also as a member of the scientific committee of map design, we are working together, map design and the SFTCG, to organize a day dedicated to innovations for cell and gene therapies, gathering again industry and academia. So I believe that it could be a very nice opportunity to continue the discussions. So thank you for your attention, and on the behalf of the team, thank you.
Thank you very much, Oumeya Adjali. Thanks a lot for this brilliant introduction. And you see that AAV vectors will be really the focus of several of the short talks this morning, and of course during the roundtable at the end of the morning. So it's very important to get this overview. Thanks a lot.